Hello, I am Biagio Ricciuti, a thoracic medical oncologist at Dana Farber Cancer Institute. I'll be reviewing a few abstracts and presentation from this year ESMO. This year ESMO has actually delivered several important advances and updates in the way we understand and treat lung cancer. And there are three presentations in particular that stood out that I'd like to review. The first one is interesting because it does explore a completely new dimension and different angle of immunotherapy response, which is the role of the thymus. In this study, using approximately 3,500 real-world patients treated with immunotherapies, the investigator applied a deep learning approach to test routine chest CT scans to quantify what they called thymic health. Thymic health was referring to the thymus size, shape, or tissue integrity in general. And the results were fairly striking because in over 1,200 patients with non-small cell lung cancer, a higher thymic health correlated with a 35% reduction in the risk of progression to immunotherapy and 44% reduction in overall mortality. And similar association was also noted in patients with melanoma, renal cell carcinoma, or breast cancer, suggesting that this biomarker, potentially clinically available through CT scans, can be transversal across multiple cancer types. More importantly, using validation cohorts and specifically PCR sequencing analysis, that did also confirm that this imaging-based signature truly also reflects T cell activation, T cell differentiation, and overall immune competence. So this data, interestingly, because they do support a very intuitive concept, which is immunotherapy relies on healthy and functional immune system, not just tumor-based biomarkers. The second study I'd like to highlight is the MDT Bridge. This was a phase 2 study which focused on patients with resectable or borderline resectable stage 2B to 3B non-small cell lung cancer. All patients in this study did receive a short course of neoadjuvant durvalumab, a PD-L1 inhibitor plus chemotherapy, after which a multidisciplinary team reassessed whether surgery or concurrent chemoradiation therapy was the best definitive strategy. And the results are fairly encouraging because 95% of patients ultimately underwent definitive therapy, whether there was surgery or concurrent chemoradiation therapy, and the overall resection rate was actually 86%. But perhaps the most impressive results was that more than 80% of patients who were initially considered borderline resectable were converted to resectable after induction chemotherapy. And this is a meaningful real-world shift that expands the pool of patients that are now eligible for curative surgery. And among those who did proceed to resection, 28% did achieve a complete pathologic response. This is consistent with what we have seen in neoadjuvant or perioperative clinical trials. Finally, the last study I'd like to highlight is the OptiTROP 4 trial. This was a randomized phase 3 study that presented highly promising data for patients with non-small cell lung cancer who have an EGFR mutation and have a cancer that has unfortunately progressed after a targeted therapy. A setting where we do have certainly good options, but somewhat the number of approaches we have for our patients remains limited. This trial looked at sacituzumab tirumotecan, a novel TROP2 antibody drug conjugate. This was compared with standard of care platinum-based chemotherapy in this pretreated patient population. In the study, sacituzumab tirumotecan significantly improved outcomes across all endpoints. The median progression-free survival was nearly doubled from 4.3 months to 8.3 months, for example, with an hazard ratio of 0.49, and 12 months progression-free survival rates, which were 4 times higher. Overall survival was also improved with sacituzumab tirumotecan, with a median overall survival not yet reached, compared to 17.4 months in the chemotherapy arm. Response rates as well as duration response were also better with the antibody drug conjugate, positioning this molecule as a strong candidate for the next standard of care for patients with EGFR mutant lung cancer who have experienced resistance to prior targeted therapy. I think together these studies capture clear momentum in our field. We identify new biomarkers that are not only based on tumor cells or tumor analysis, but actually rely on the host immune system, for example, therefore moving beyond PD-1 expression or mutational burden. We are refining multidisciplinary strategies to expand the curative treatment for patients with locally advanced disease in an area of intense research today. We're also introducing next generation antibody drug conjugates that are capable of delivering meaningful benefit in molecularly defined subset of patients, such as the EGFR mutant non-small cell lung cancer population.